I am Andrea Perez, and I have the amazing opportunity to be working under the mentorship of Dr. Fudasi from the Chemical Engineering Department. Dr. Fudasi works with an amazing research group who is helping him find an answer to his main question. How can we make better and more efficient separation processes for environmental and biomedical processes? On the search for an answer, his research group is working on developing membranes with better filtration performance. These membranes can also be used for protein purification and bioprocesses in the pharmaceutical industries. The second project is the production of hydrogels for better water absorption. Hydrogels are also used in scaffolds in tissue engineering. The third project is the separation of exosomes from other cell components. My part in answering this question consists of making membranes capable of efficiently separating exosomes from body fluid samples. Quick question. What are membranes? What are exosomes, and why do we want to separate them from body fluids? Awesome questions. Now, a membrane is a thin layer semi-permeable material that can selectively separate solids from a medium. Now, exosomes are microvesicles which are secreted by both normal cells and cancer cells. They are distributed in many body fluids such as blood, saliva, and urine, and they can carry many types of biomolecules. During cancer cell development, cancer cells secrete more exosomes with significant change in composition. These facilitate communications with the tumor environment, drug-resistant acquisition, and metastasis to distant organs. We want to use membranes to efficiently separate exosomes from other components in a fluid sample for cancer diagnosis. Let me show you with a visual representation what I mean by separating exosomes from body fluids. Welcome to my lab. I usually use it to make cookies, but today I'm going to show you a visual representation of my project in exosome separation. Now, let's get our gloves on. Now we're ready. Now let's pretend that this is your cell. As I said before, cells secrete exosomes. Now let's see what an exosome looks like here. Here. This is your exosome. But cells don't only secrete exosomes, they secrete other biomolecules. For example, in here, pulp, right? Okay. Now let's pretend this colander is our membrane. As I said before, a membrane is a thin layer of semi-permeable material that can selectively separate a solid from a medium. Now here, we have our body fluid sample. It can be blood, saliva, urine, whatever. And in here, my job is to create a membrane that can efficiently separate the exosomes from the other biomolecules in the body fluid. Sorry. Like this. As you can see, the membrane not only separated the exosomes, but it separated other kind of biomolecules. Now my job is to create a membrane that can efficiently separate only the exosomes and leave the other type of biomolecules behind.